Dr. Junhao Wen is the principal investigator of Laboratory of AI and Biomedical Sciences, or Labs. He is an assistant professor in the Department of Radiology at Columbia University and holds affiliated appointment in the Department of Biomedical Engineering at Columbia University and the New York Genome Center. He is also a visiting professor in the Department of Radiology at the University of Pennsylvania. Right, and you can please take it away.
Thank you. Thank you very much for allowing me to present my research at the department. So the topic of my talk today is modeling human aging and disease at scale, AI machine learning, imaging, genetics, and beyond. So as I'm new to Columbia, I would like just to briefly introduce myself for my research career trajectory. So I did my bachelor and master in electronic engineering at Beihang University in Beijing, China. And after that, I went to Sorbonne University in Paris to pursue my PhD in computer science. During my PhD, I also had a chance to work as a visiting scholar at UCL in London, working on diffusion MRI stuff. After my PhD, I came to the United States, working as a postdoc at University of Pennsylvania in computational neuroscience. In last year, I went to University of Southern California as a tenure track assistant professor, and then I decided to come back to the East Coast. That's how I ended up at Columbia. So for today's talk, I'm just gonna briefly go over my research trajectory, to give some personal perspective regarding personalized medicine. It's going to be a personal version of AI in biomedical and clinical neuroscience. There are three keywords here that I want to highlight. Basically, the three main projects from my PhD postdoc and towards my independence to work on, to advance precision medicine. The first one is reproducibility. The second one is heterogeneity, and the third one is multi-scale modeling. So I just want to briefly summarize my research topics from the lab. If I have to put one sentence to summarize this, those projects, I would say AI and machine learning can see what we don't see as human beings. I'm pretty optimistic regarding the application of AI and machine learning. With that being said, I'm not claiming here at all that those AI and machine learning masters can replace the critical role of clinicians like neurologists, radiologists, etc. And there are several key aspects. The first one, the keyword here is multi-omics. I was trained by a neuroimaging machine learning. Sorry. Okay, so I was trained by neuroimaging and machine learning AI, but during my PhD postdoc and towards my independence, I want to develop some new directions or new methods. That's when I have been interested in bringing other omics data into my research. This include genetics, genomics, proteomics, metabolomics, and potentially in the future maybe single cell resolutions for those omics data. And of course, the core part of the work in the lab is to develop and apply those various AI and machine learning methods, including conventional statistical methods, conventional machine learning methods like SVM, and more cutting edge deep learning methods like neural networks. Hopefully, those methods can be applied to different applications, including disease classification, prognostic, and clustering, which may benefit downstream patient care for drug development, for example. Another key aspect of the research here is to modeling human aging and disease at multiple scales. And this scale can span a different organ system, of course, but also at omic data types. The first project I would like to briefly go through is focused on reproducibility in neuroimaging community. And I think when I started my PhD at that time, Nature has this is after my PhD anyway. So Nature has asked the community whether there is a reproducibility crisis in the imaging different domains like neuroimaging, machine learning community. And surprisingly, we can see the community has a very has been very cautious about this reproducibility crisis. And specifically for my PhD topic, we have been trying to benchmark reproducibly and objectively using machine learning and AI methods to classify Alzheimer's disease using neuroimaging data, brain MRI scans. And we have been provided open source software. We have provide like codes to convert the publicly available data sets in a reproducible fashion. So researchers can directly use those like framework and software to try new algorithm, for example. And when I start my PhD at that time, of course, as a new guy from to the imaging field, I didn't know much about like Alzheimer's disease classifier, how the accuracy is in the field. So I did a literature review just to check the state of the art. And surprisingly, a lot of as a table shown here, the top part is a paper actually who might have like potential like data leakage or methodological flaws. In those papers, they reported like nearly 100% of accuracy to predict Alzheimer's disease from healthy control. So at that time, I was thinking, okay, if those paper has already done so good, why should I? What should I do for my PhD? How can I improve those things? But if you check those paper like carefully, you might see a lot of like imperfect or unperson like or somehow they have like some methodological flaws. And the table on the lower part basically showed those papers that we carefully scrutinized, and we didn't like identify any evidence for methodological flaws. This basically highlight the reproducibility crisis in the specific domain using neuroimaging data to predict Alzheimer's disease. I will introduce like some specific scenarios why those like over-optimistic results may happen. So to demonstrate our argument that this reproducibility crisis happens like in neuroimaging field, we used like 3 publicly available data sets for Alzheimer's disease, ADNI, ABO, and OASIS. And we benchmarked those data, MRI data, using different CNN architectures to predict Alzheimer's disease using convolutional neural network to unbiasedly benchmark the accuracy. We split the data into training validation for nested cross-validation in order to select the optimal model. And we also hold out an independent test data set. And we didn't touch the test data set until the second round of peer review. We submitted paper only for the first round, only for the training and validation results. After the reviewers were happy with our architecture, we applied the training model to the independent test data set. So to make sure the model was not overfitted. We tried different architectures regarding the dimensions of input imaging. 3D, like if we put like 3D the entire brain MR scan, and also we extract some specific region like hippocampus region, which is a home marker like for Alzheimer's disease. And of course, we also split like the 3D imaging into 2D slices and then fit into a convolutional neural network. We have answered several questions here. The first one I want to highlight is we always wonder is whether a deep learning model like CNN works better than conventional machine learning methods like linear SVM. So the conclusion, at least from the data what we have here, almost 2,000 people per MR scan for Alzheimer's disease, the answer is like they perform quite like comparably. With a linear SVM classifier, we got like balanced accuracy for 0.88, and the best CNN model we got from the 3D hippocampus region-based CNN. And of course, there are many like factors that could contribute to this phenomenon, potentially because like we don't have enough data set for the Alzheimer's disease classification for neural network. And the second question we were trying to answer is to answer whether the model trained on one data set will be generalized to the other like independent data sets. So the model was trained on the ADNI, which has the largest sample size, and we test whether the model can be generalized to ABO and OASIS. And as we can see here, using 3D subject convolutional neural network, we can see the model was generalizable overall to ABO data set, but not OASIS. And the underlying reason, of course, may be caused by domain shift because of the demographics differ quite a little bit between ADNI and OASIS. And also, I think in OASIS data, they put like mild cognitive decline into the patient groups, which is not the case for ADNI. Finally, we demonstrate like a specific scenario where like why the previous paper that I mentioned in the table reported like very high accuracy. And to demonstrate this, we use like 2D slice input data from the brain MRI scan. And the figure on the top showed basically when we split the data, if we split the data at the slice level, which means when you split training and validation, the slices from the same people, they can go into both training and validation. Basically, that's kind of data leakage because the slices from the same people, they're highly correlated. And that's where you get, okay, the training and the validation almost 100% accuracy. But if you have a holdout independent data, test data set, when you apply the training model to the test data set, you can see a huge jump to 0.76. The figure below here differs from the previous approach. Here, we split the training and validation data based on the subject, not on the slices. That ensures the slices from the same people always go to one data set, either training, either validation. And by doing that, we can already see a model jump, like overfitting phenomenon from the training to the validation. And of course, it's not going to generalize very wide to the independent test holdout data sets. So we made, like I mentioned earlier, we made all the converter for the 3 data sets, image processing, machine learning models from scikit-learn and PyTorch, publicly available. And I'm like, until now, I'm still very proud of this project because I think this has some impact to the field. So the second project is basically my postdoc work at UPenn. And at that period, I have been very interested in using like AI and machine learning methods to understand disease heterogeneity. Because we know, like clinically for Alzheimer's disease, people think it's mainly a like brain disorder, but within the patient population, you can clearly observe like heterogeneity regarding clinical symptoms, cognitive scores, genetic, like SNPs, genetics genes, and of course, like different brain atrophy patterns. And that's the main motivation for my postdoc work. And we are trying to using like neuroimaging data to find like more homogeneous subtypes, what we call the dimensions of Alzheimer's disease. And of course, this we have been like this at the end of the day, this is a classroom method from like machine learning perspective, right? Our approach differs quite a bit from like the most common like classroom methods. For example, unsupervised learning like k-means. Basically, you just fit all the imaging features from all the patients into the algorithm like k-means. It operates directly in the patient domain and give you some clusters or subtypes. Instead, what we are trying to model is not what we call like semi-supervised learning on the like the figure shown on the right side. We call it like semi-supervised learning or weakly supervised learning. So basically, we are trying to model what we call the 1-to-K mapping from healthy control, the reference group, to the patient domain. So that's, in our opinion, it's like more clinically plausible because Alzheimer's disease, for example, progress from healthy control to mild cognitive decline to Alzheimer's disease. By doing this, we kind of like to extract the like etiologically or pathologically related variants only, like excluding other confounding factor like demographics. Like in line with this principle of the semi-supervised learning framework, we have developed like several different methods. More recently, we have using like generative adversarial networks to model this heterogeneity, like particularly for 3 models called SmileGAN, SerilGAN, and GeneScan, which I will briefly introduce later. And besides those methodological development, we have been also very interested in like applying those methods, of course, to clinical population, try to understand specifically like clinical insights within different brain disorders, like little life depression, autism spectrum disorder, and also like typical aging. I will just briefly go through the magic model, which I developed during my postdoc. And this is an extension from our previous model called Hydra. The intuition here is quite straightforward. So following the principle of the semi-supervised learning on the figure B here. So basically we use like multiple linear SVM to construct a polytope. This polytope basically separates the healthy control and the patients. By doing this, you can assign the patient to the nearest face of the polytope. That's how we construct this 1-to-K mapping. And the novelty here, of course, it's also actually to adapt a brain feature extraction spanning like at multiple scales from 32 like brain population to 124 population. And we have shown actually by adopting this multi-scale modeling feature extractions, it outperformed like the original single scale features via Hydra. And then we apply the model to, yeah.
Okay. When you say you did a SVM, I think you referred to some previous results as well, and it was very good. And what is the input? Is this like the full 3D image that is being run through? Like what is the actual input that goes into generating these like sort of polytope boundaries?
So that's a good question. But the previous, when I mentioned linear SVM, that's for classification, right? That's to classify RSM in patients. It's quite straightforward. You can, I mean, SVM has been known to very robust to high dimensional imaging features. In that case, you can even fit like the 3D MR scan, which has millions of voxels. Yeah. It has been shown like very robust. Here, it's similar. So basically the polytope here perform both like the classification and clustering, but instead the subtype is defined, or the cluster is defined by assigning the one specific patient to the nearest polytope and face of the polytope. So the features here, you can of course fit like 3D MR scan, but we have tried that. Actually, it's very computationally heavy, and second, the polytope is not very stable when it cannot deal with the high dimensional features in this case. So in this case, we fit like the ROI like regional brain volumes, briefly like 200 brain MRI brain regions. Does that answer the question? So here are some clinical applications using those machine learning AI methods we developed in the lab. In this paper, we have applied like the magical model to a clinical population of late life depression consolidated from various resources. And first, we run the algorithm, find two dimensions or two subtypes. Sorry. So the first dimension basically showed there are no like brain atrophy pattern of the brain. Instead, we observed like larger like brain volume in the subcortical structure, which is a little bit surprising because in the literature, if you run a case control study, but most of the neuroimaging study found like global brain atrophy pattern, which is shown on the dimension 2. Like for example, brain atrophy in the bilateral cortical regions. I think it's very interesting finding for the dimension 1, why a small proportion of the patient population have like the preserved brain volume in the subcortical structure. And then to answer that, we run a genome-wide association, and we found like one specific loci here, which might show like protective effects. But we need to be cautious about like to interpret those results here. First, the sample size here, I think it's not very big, like around like 2,000 people. It's very small for GWAS studies. And second, I think the signal here is not very strong in the sense that only one specific SNP that passed the genome-wide association threshold. And of course, GWAS is just association, it's not causality. And then we also like try to understand at the microstructural level using diffusion MR model, try to understand why the two dimensions differ from white matter interruptions. And we found the dimension 1, which did not like show any white matter disruption. And in contrast, the dimension 2, which showed brain atrophy, also showed white matter disruptions over the entire brain. The next question we are trying to answer is whether those late life depression dimensions will serve as a risk factor to develop RSM disease. Because in the literature, late life depression has been a very high risk factor or comorbidity for developing like RSM disease in the old population. To answer this question, we use ADNI data sets, and we apply the training model to the ADNI longitudinal data sets, which has 7 years follow-up. The first question is we were trying to answer is why those two late life depression dimensions remain stable along the 7 years follow-up. And on the figure, on the top part of the figure, as we can see, the two dimensions indeed remain like quite stable. That means the two dimensions do not intertwine too much with each other along 7 years or 7 years follow-up. And the second question we were trying to answer is which dimension is more accelerated longitudinal speaking to RSM disease and to aging. And as what we can expect, of course, like the dimension 2, which show the global overall like brain atrophy pattern will develop quicker compared to the dimension 1 to aging and RSM disease. Another example of applying those methods to ASD, autism spectrum disorder. This is a collaboration between between me and another postdoc when we were at UPenn, and we applied the methods to a population of ASD, and we found like 3 subtypes or 3 dimensions here. Similarly, we applied the training model from the ASD population to the UK Biobank general population and try to understand, because the sample size are larger, and we try to understand what genetics will affect what specific gene will affect a specific dimension of the disease. I will not go into the details due to the time. Another part of the research here, we were trying to, like I mentioned earlier, those methods I mentioned earlier was like based on conventional machine learning methods like SVM. Here, another branch of research, methodological development, has been done by the generative adversarial network. This was done by our previous PhD student at UPenn that worked with me closely, and we developed like the SmileGAN and SerialGAN gene scan. So SmileGAN here is very similar. The concept is very similar with the magic of the Hydra. Basically, we were trying to using GAN, the GAN part, to model the 1-to-K mapping via a generative approach. And the gene scan basically is an extension on top of the SmileGAN by ensuring like the subtype or dimension to be simultaneously linked to both imaging features and genetic features. Because all the previous model I was talking about is based on the cluster is based on solely on imaging variance. And SerialGAN here it slightly differs from the 2 models here in the sense that the underlying assumption is slightly different. So SmileGAN has been always trying to find a dominant like subtype in one patient, which assumes the patient only shows like one strong patterns. So the SerialGAN here is a representative learning method, slightly different from SmileGAN, which assumes like even within the same population, it can have like high dimensional scores here. In this case, R1 and R2, for 2 different atrophy patterns, for example. That's basically the small like difference between these 2 methods. Then we apply the SerialGAN model as a dimensional representation learning to a large-scale aging population. In total, I think over 50,000 people. And we define those population as pre-aging, I think people younger than 40 years old, and after aging, like after 40 years old. And then we apply the semi-supervised learning method SerialGAN, and we found actually 5 different patterns. That basically means in the aging population, we can expect, based on the algorithm, people can age with 5 different distinct pathway or atrophy patterns. And here we don't use any like RSM disease related information, but surprisingly or expectedly, like the dimension 2R2 basically captures the medial temporal lobe region atrophy and hippocampal region. That's if after that we run also a phenotypic association, and we indeed found R2, this dimension is linked to, for example, amyloid and tau, which is a hallmarker of RSM disease too. And then we also run GWAS analysis and found like gene linked to RSM disease, which are enriched in the R2 MTL atrophy patterns. So the third research topic I've been interested in it's more like my current research. Like I mentioned, my previous research has like primarily focused on the brain, because RSM disease has been considered a brain disorder, and also imaging data. And we brought in a little bit like omics data here too. So towards my independence, I want to do something different from my postdoc lab. So that's where I have been interested in bringing like genetics, other omics data into my research. So my current research has been inspired largely by many papers, but I just want to highlight 2 papers here. The first one, I think it's the first large-scale brain imaging genetics study from Elliott and colleague, published in Nature 2018. And after that, I think there are many papers trying to linking like brain imaging derived phenotypes versus like a common genetic variants. And right now, I think people are also trying to link to rare variants too. And another paper from my collaborator at UPenn, Bingxin Zhao, who published this Nature Science paper last year, trying to link the brain MRI and heart MRI with underlying genetics. So, but my approach might differ a little bit, because those 2 papers look at like the conventional brain atlas-based imaging features, But what I'm trying to do here is trying to define those new phenotypes defined by AI machine learning algorithm. One specific example here is, like I mentioned, all the two papers I mentioned earlier, they define the brain postulation based on some existing atlas, like frontal lobe region and temporal lobe region. Here, we are trying to run a signal projective non-negative matrix factorization, try to parse like the brain into what we call patterns of structural covariance. Basically, it captures the within voxel and cross-voxel structural covariance between different individuals. And the good part of the NMF-based methods compared to PCA, as we all know, it's that it will give you like part-based brain regions, and instead PCA will give you very diffuse over the entire brain, which is not very good for clinical interpretation. Here we apply what we call OPNMF. We add additional signal constraint to the NMF algorithm and postulate the brain at multiple scales, from 32 to 1024. This is actually the feature that we use for the magic model, that I mentioned earlier. And the overall hypothesis here is that we define those phenotypes using machine learning, which is like purely data-driven, and we hypothesize that those imaging features might provide higher statistical power to find like new genes, new genomic loci linked to underlying genetics. To prove that, we run large-scale GWAS analysis using more than 50,000 people from the ICD consortium. On the figure A shown here, actually we found indeed compared to previous conventional imaging feature-based GWAS, we found more genetic signals. Like I mentioned earlier, GWAS is just basically an association, right? If you have a large sample size here, in this case, UK Biobank, you have more than 40K people, you can definitely find some signals. But the question here is how to validate or how to interpret like those genetic signals. There are like 7 different approaches to do those post-GWAS analysis. I'm just gonna showcase some analysis we have been doing in the lab. The first one is, of course, to do gene set enrichment analysis to interpret like enrichment biological pathway linked to the different genes of those patterns. I will just explain, like showcase like 2 examples here. On the figure on the right side, for example, it's basically captures the medial temporal lobe region, which is a hallmarker of Alzheimer's disease. And we run GWAS. Then we map those SNP signals to gene. Then we perform gene set enrichment analysis, and we found actually this has been implicated or enriched in several biological pathways of Alzheimer's disease. For example, the nuclear membrane, which is a biological pathway, it's not unique for Alzheimer's disease, but also has been like implicated in AD. And another example is for the imaging based machine learning based phenotyping discovery is that we use AI machine learning to predict chronological age using brain MR scan, which we normally call the brain age gap, brain age, for example. And here we use the data from UK Biobank and 3 different brain modality: gray matter from T1 weighted MRI, white matter from diffusion MRI, and functional connectivity from resting state functional MRI. And we benchmark the age prediction performance using the 3 modality and define those 3 AI-based brain aging phenotypes. Then we run the GWAS. After GWAS, we run the PHEWAS, basically linking the genetic signal back to the phenotypes. In another study, which is currently actually in press in Nature Biomedical Engineering, we also investigate further the disease subtypes that we found in our previous study, those with our semi-supervised learning disease methods. Here, we kind of look at those disease heterogeneity as a whole, which means that now we define those subtypes for Alzheimer's disease, which has 2 dimensions or 2 subtypes. We found previously 3 dimensions for autism spectrum disorder, 2 dimensions for late life depression, and 2 dimensions for schizophrenia. And the interesting part is that those subtypes were defined by purely imaging variance. But if you compare those subtypes, our dimensions across the different brain disorder, you see a lot of overlaps, similarities, and that motivated us. Okay, can we find like some common genes, our shared genomic loci that simultaneously shape this brain atrophy pattern across different brain disorders? And to do that, of course, we run GWAS analysis, post-GWAS to validate those signals. And we also linked those dimensions to proteomics features at different molecular level, proteomics data and metabolomics data too. Like I mentioned, I will not go into details, but if you have any questions, I'm very happy to answer. So our previous study here, we still focus on the brain, right? So like I mentioned, Alzheimer's disease has been considered as a brain disorder, but like competing, like the main hypothesis is amyloid and tau hypothesis, the cascade amyloid and tau. But even in the field, I think people are also discussing alternative hypotheses that linking potentially other organ systems like immune, cardiovascular, comorbidity, all those things. That motivate us like try to model like those aging and Alzheimer's disease at multiple scale across like different organ systems. In line with this research topic, we have published like several methods. For example, one method, we'll just go there. So this paper actually just basically extends the brain age paper we published previously in total 9 different organs. Right now we have the other organs like cardiovascular system, musculoskeletal system, and retina or IBAG. We did similar analysis, define those phenotypes using machine learning AI methods, run GWAS, post GWAS. Here, just another use case how to validate those GWAS signal. Here, we perform 2-sample Mendelian randomization, basically try to using the SNPs as an instrument variable to mimic randomized clinical trial, try to understand the causality between those non-brain age gap, biological age gap versus different human diseases, for example, and also lifestyle like sleep and so on. Just try to highlight some specific signals here, like as we all expected, the Alzheimer's disease positively affect the brain age, which makes sense, right? If someone have Alzheimer's disease, it might increase your brain age gap. If you have a higher brain age gap, actually also cause your sleep duration. Because if you have a higher, which kind of makes sense, you have higher BAG age gap, you look older, your brain looks older than your true age, and that could affect also like your sleep duration. Another example is like body weight, which we all know is very important and implicates the overall systemic health, and it has been causally linked to different organ systems. But like I mentioned, when we interpret those results, we need to be cautious because Mendelian randomization is a statistical method with a lot of underlying assumptions, and oftentimes it violates underlying assumptions. In the paper, we have done like multiple sensitivity checks, but like I mentioned, we should be cautious to interpret those results. Another example, try to kind of validate those genetic signal, to answer some neuroscience questions here, is that in this paper, we also try to verify the Chevreul conjecture. This conjecture basically hypothesized like if you don't have the power to collect large-scale phenotypic data, because maybe phenotypic data collection is more expensive, but if you have genetic data instead, basically you can estimate the two clinical traits, their genetic correlation, and this genetic correlation can be used as a rough like estimation for the phenotypic correlation. And the figure shown here is we calculate the pairwise genetic correlation between the non-biological age gaps for the non-organ systems and on the upper triangle part. And the lower triangle part basically, the upper triangle part is the phenotypic correlation, basically a Pearson correlation. And lower triangle part is a genetic correlation. As we can see here, basically it supports like the Chevreul conjecture, which basically means the genetic correlation of two traits basically mirror their underlying phenotypic correlation. A new study we published like recently is to leverage the GWAS summary statistics to better understand the cross-organ and within-organ interactions here. The good thing I think about the genetic research is that a lot of those markers have been developed using only the summary data, which do not require the individual level data, because we know like genetic data is very sensitive regarding data privacy. And here we try to use genetic GWAS study, a summary statistic developed by our team using UK Biobank, and with other summary data from the FinGene and Psychiatric Genome Consortium data sites. And we found like the within organ and cross-organ interactions. Finally, I just want to highlight, I'm a big fan of public dissemination for research funding, and also secondary derivatives. This includes like pre-trained AI models, and also like GWAS summary statistics. We have developed like 3 public portals to disseminate our research, and hopefully this can benefit the community for their own like analysis. Finally, I just want to acknowledge the help from our collaborators who can greatly contribute, like the MART Consortium from Penn, from NIA, University of Melbourne, USC, and the ICL. Thank you very much. I'd be very happy to answer any question.
All right, if anyone
If anyone has questions on the Zoom, oh, I see a hand already. Abby, if you want to just unmute yourself.
Yeah, thank you so much for your talk. My question is about just examples that you gave throughout the talk, I guess, in which patient subtypes are identified by machine learning approaches and then subsequently validated through GWAS. And so I'm just wondering if you could talk a bit more about how confounders are handled in these examples, like demographic variables. So I guess essentially, are these confounders adjusted for within the machine learning method during the GWAS included as covariates, or sort of both?
To directly answer your question, I think both at different stages. For example, I'm just going to take this project as an example. So the model, the full model, like for the full population, Alzheimer's disease, ASD, late life depression, schizophrenia, they were developed, the original model, they were developed like in the disease-specific population, right? Using like the AI machine learning methods to define your subtypes or dimensions. But those original data set does not have like genetic data, which does not allow you to run large-scale GWAS. To do this, we have applied the data to the UK Biobank, which demographically and also age range, they differ quite a bit from the original training model. And we handle this to a certain extent. For example, we statistically harmonize the imaging data using the original healthy control from the full population as a reference, basically map the brain volume from UK Biobank to the original sample, and then we apply the pre-trained model. I think we use like very simple like linear regression models, but ideally, I think there still has some like domain shift due to different like the age range and other like age and sex, all those things. And when, of course, like you mentioned, when we run the GWAS in the downstream analysis, like in the UK Biobank, we always include like age and age squared, the interaction between age and sex and other like genetic PC, all those covariates to correct for those confounding. So yes, we do care about like those confounder as a covariates, but we all know it's not sufficient like to handle this potential domain shift.
Thank you so much.
Thank you.
Okay, and I think we have a question on the chat from Maureen. Says, on slide 26, it's interesting to see a positive arrow from IBD to musculoskeletal. Could you please elaborate on that?
IBD to musculoskeletal, that's a positive causality, which means if you have a IBD disease diagnosis, you might have higher musculoskeletal BAG. A higher musculoskeletal biological age gap means your musculoskeletal system looks older than your true age. I think this should be expected, no? If you have the disease, your disease related to IBD, I don't know, I mean, I'm not very familiar with, expert with IBD, but if you have certain diseases that might affect your musculoskeletal systems, that makes your musculoskeletal age looks older. Does that make sense?
Does that answer your question, William?
yes.
But I agree. Actually, when the paper was under review, we do get some like counterintuitive regarding the direction of the causality. For example, someone would expect, I think another example here maybe we should talk about is Alzheimer's disease with a hepatic, for example. If you have Alzheimer's disease diagnosis, this is positive. But anyway, we found some weird like directional causal effects between the aging clocks with regard to disease. I think the problem for GWAS is that, like I mentioned, it's very sensitive regarding the selection of your underlying instrument variables. Even though you consider LD, you consider like the highly correlated SNPs, sometimes it's not the golden standard. The golden standard of the ground truth is going to be you perform a randomized clinical trial, which is super expensive. And I don't think right now it's like available. That's also why I mentioned like when we interpret like those results, we need to be cautious.
And if you want to just unmute yourself.
Oh, hello. Thank you so much for the presentation. I was just wondering, like, if there are methods you use to kind of assess like the quality of the data that you use and like how you select the kind of samples to be included in these types of like large scale analysis across multiple systems.
Sorry, can you repeat your question? I don't quite get it.
Yeah, so I was just wondering like if there are methods that you use to assess like the quality of the data that you used to be included, since you mentioned that it is kind of difficult to assess like whether these causal relationships are valid, and you have to be careful with the kind of conclusions that you derive from these studies.
I'm not sure if I understand it correctly, but do you mean like how we handle like those confounding factors from different diseases of the population, or how we select those populations?
I think more about how you select the samples to be included.
So like in Mendelian randomization here, you use GWAS summary statistic, right? Depends on the traits or phenotypes. For the BAG, for example, basically we include population from the entire UK Biobank data. UK Biobank data is not a disease-specific data; it's a general population. They might have like disease diagnosis from inpatient, outpatient services at a hospital, like defined based on the ICD code. We include those diagnoses as a covariate in the GWAS. Hopefully, can correct those confounders. But for example, for the disease endpoints here, like Alzheimer's disease, like type 2 diabetes, we use the existing GWAS from other resources, because Mendelian randomization needs the exposure and outcome. They don't have like overlapping population, so we cannot use like for example the GWAS from UK Biobank for Alzheimer's disease. And I think that's a good question, because in the field recently there are paper published in Nature Genetics. People like geneticists, when they perform the GWAS, they try to increase the sample size, because they assume with a larger sample size you get more signals, you get more robust signals. But the problem for Alzheimer's disease is that in a clinical setting, you define Alzheimer's disease based on the clinical, like the cognitive scores, imaging biomarker or plasma biomarkers. That's very like sensitive and accuracy. I think today Nature Medicine published a Nature Medicine published paper using like plasma tau, a variant of the tau, like achieving like 90% of accuracy, which is pretty good in clinical setting. But those GWAS, they define those disease status in a very broad way, which could introduce a lot of confounding factors for downstream associations study, because they want to get more cases. They might define AD case here using like proxy by, if your parents get Alzheimer's disease, they put like this, they define this AD by proxy and treat it as an AD patient, which actually doesn't make sense, because most of the spectrum sporadic AD is not dominated by genetics. So in that case, yes, there are a lot of confoundings between those exposure and outcome GWAS that we use this study. But I think that's a very general like challenge or question in the field, how to battle against like those sample size limits and confounding factor from the population, from the diagnosis. I hope that answers your question.
Yes, thank you.
think there's another question in the chat. It says, from William, can you further clarify the arrows around with weight, especially the double arrow with pulmonary? What does a positive arrow indicate? What does weight represent, overweight or underweight or both?
The weight here, I think it's the overall weight, and the negative sign means the odds ratio, basically the SNPs, the independent SNPs from your weight GWAS and the SNPs from your pulmonary aging clocks. Let's see, we define those 20 SNPs as instrument variables, and if you correlate those SNPs from the two GWAS, body weight and the pulmonary aging, it's negative, which means that it's bidirectional because we switch the weight as exposure as outcomes. And to interpret the negative association, for example, when from the pulmonary aging clock to the weight, in this case, the pulmonary aging GWAS as an exposure. If your pulmonary aging increase, the gap increase, which means your pulmonary aging looks older than your chronological age, you have lighter body weight. I don't know if that makes sense, totally makes sense or not. That means if your pulmonary system looks older than your calendar chronological age, you might lose weight somehow. Does that make sense?
Got it. Does that positive arrow from weight to renal, that means if you have overweight, your renal will look older than your chronological age?
Yes, that's correct.
Okay, gotcha.
If you're overweighted, you might, like your renal aging clocks might look older than your calendar age.
Got it. Thank you.
Again, all those conclusions are from statistical perspective. So we need to interpret those results with caution.
Are there any other questions?
Well, and just to point out, I mean, being underweight as you age is not always a good thing, right? So I think just pointing that out clinically as you're talking about these relationships, the assumption that overweight is not good, it can go the other way too as you age.
Yeah,
which I think speaks a little bit to a potential hypothesis for that.
Another thing we consider in genetics, it's like pleiotropy. Basically means the genes can affect different traits, like your body weight, your other organ system. Here we try to exclude those SNPs with pleiotropic effects. So we exclude them, but still, I mean, the valid SNPs can still exert like pleiotropic effects, which might, the body weight might affect another trait that the trait influence your renal aging systems. We don't know.